I know. I would blow up the whole room before I'd let that thing out of here. I'd be like, you're all failing. Give me that recording. Okay. So we're going to finish this set of notes, and then we are going to start the next one. Today is one of those days where you really, really, really need to ask questions when you get confused, because this stuff is really fundamental for the next bit of material, especially once we start talking about the electrical nature of the cell membrane. So if you don't get it, you are not the only one, and I promise I will be kind in helping to go through it again. Yeah, kind of. I mean, it involves that for sure, but not exclusively that. So the last thing that we were doing was the sodium-potassium pump, right? Yeah. And I told you guys that the animation was posted on the links page, but it's actually a fib because they took it off. So the only way that you can see it that I could find is through my video, because I don't have copyright permission to post it. So you're just going to have to see it through my video. It's owned by McGraw-Hill, which is a different textbook than yours, so they won't give you access to it. So it's on my video there. Other than that, you can't see it. The link won't work.
Melanie, are you recording?
Last time we did the sodium-potassium pump. And before we get started on the new material, I want, instead of me teaching you the whole cell and how it gets charged and everything, I want you guys to teach me. I want you guys to sort of slowly work me through it, because it's going to be sort of fundamental to the rest of the stuff we do today. So, get my pen. All right. So I'll tell you, we're just going to start out the way that we always do, with the ICF and the ECF. Okay. And we are first going to ask you the positive ion that's in highest concentration in the intracellular fluid. What is it? Intracellular fluid.
Potassium.
Yeah. High concentration of potassium, right? And a relatively lower concentration of potassium here. Yeah? Mm-hmm. Okay. Do you remember what the ratio is?
3 to 1.
No, it's 30
31.
to 1. Okay. And as far as sodium, where is sodium in higher concentration? Yeah, sodium is in much higher concentration in the extracellular fluid and lower in the intracellular fluid. Okay. What's the concentration gradient here?
1 to 15.
Yeah, it's 15 outside to every 1 inside. Okay. And so what we're going to do now, and then we'll sort of mess with it later, so this is kind of fundamental that you understand, is we're going to put in leak channels for sodium and potassium, just like we did last time. Okay? Let's make this one a potassium leak channel. And this one a sodium leak channel. Oops. And the question that you know the answer to is, which direction are they going to leak? Which direction is potassium going to leak? Out. Which direction is sodium going to leak? In. Okay, so we've just created a problem, because this is homeostatic and this is homeostatic, yes? So the leak channels create a problem, and how do we solve the problem? What? Yes, sodium-potassium pump. Yes, the sodium-potassium pump. And the sodium-potassium pump pumps what out? Sodium. And what in? Potassium. And uses what to run the pump? Okay. And used ATP as ADP. Okay, so we'll deal with the quantities and the leak rates in just a second, but you've got this part down. In the absence of the sodium-potassium pump, what do you predict would happen about the sodium and the potassium concentration gradients? In the absence of the pump. What would happen to your concentration gradients?
Oh, without the pump?
Without the pump.
They would go off. They would be completely off.
What would happen? Yeah, they just reach, you wouldn't have a concentration gradient after a minute, right? In the absence of the pump. Does that make conceptual sense to you? Okay, so now what we are going to just acknowledge is that this pump exists in every single cell in your body, okay? And in order to maintain homeostasis in every single cell in your body, this pump is constantly working, right? Otherwise, you would not be able to maintain homeostasis. In cells that do massive disturbances of the concentration or electrochemical gradients for these ions, like on purpose, and there are cells, then you're just going to have to use the pump more frequently, right? So you're going to end up gobbling up tons of energy. As a matter of fact, per cell, approximately 40 to 60% of the ATP that is used per cell at rest is used on your sodium-potassium pumps. So they are incredibly energetically expensive, and they're massively important. And we're about to see the first instance in which you are using all of this energy to your advantage. And that's the next one we're going to see right now. So before we get to that, let me see if you can conceptualize this thing. So what the pump is doing is, in addition to maintain the concentration gradients, do you remember that initially, before we actually had a charge, right? Then which one moved faster, sodium or potassium? So what we're trying to do is tell that first part of the story in which I had no leak channels and then put them in, right? And then the leak channels just leak the ions down their concentration gradients. If there was only concentration gradients to deal with, then which ion initially leaks faster? You're only dealing with concentration gradients, right? Before you get a charge, which ion would leak faster and why?
Potassium.
Potassium and why? Higher concentration gradients. So put in the leak channels, let it leak for a second, and then potassium leaks out faster than sodium leaks in, and then tell me what happens to the charges. Outside went from neutral to positively charged, yes. Okay. And the inside went from neutral to negatively charged, yes. Okay. Okay, so now homeostasis for the cell not only means that you have to maintain the concentration gradients, you also have to maintain this electrical gradient. So the sodium-potassium pump is going to be responsible for doing both of those, right? Because if you lose the concentration gradients, won't you lose the electrical gradients? Right? Okay, so what happens is the pump is actually going to set up and maintain, listen to this word, a transmembrane potential. And the transmembrane potential is the potential for ions to move across the membrane. Do you see that as long as I have this separation of charges, I have set up the potential or the motivation for any ion to move across the membrane when given the opportunity? Do you see that? Now that I have a charge, haven't I just created a reason that ions might want to move transmembrane? Yeah? Okay, so that will be important because quite often we want ions to move across the membrane and we want them to move quickly, okay? And so what I'm doing here is I'm working to make that happen. So keep that in mind. Okay, so now we're going to see for the first time one of the reasons that we spent all of this energy to do that. So we just did the pump, the sodium-potassium pump is an example of what we call primary active transport. But now we are going to look at a transport mechanism called secondary active transport. And with secondary active transport, what you're going to do is two transport mechanisms in series, 1 and 2. The first one doesn't require ATP and the second one does. Hence, secondary active transport. Do you see? Okay, so what I'm going to do in this situation is, let's see. Okay, so stick with me for a second here. What I'm going to do is, let's say I want glucose inside the cell, yes? But glucose is already in relatively high concentration inside the cell. I could pay for it, right? I could pump it inside the cell. Because if the cell was like incredibly energetically active, just because I had glucose inside the cell didn't mean I didn't need more, right? So I could pump it in, but there's a cheaper way that you can get it in. And the cheaper way that you can get it in is that you can move glucose through a carrier and use sodium's electrochemical gradient as the driving force. Okay, so What I can do in this situation is I can say, hey, sodium wants to go inside the cell anyway, right? And instead of letting sodium just move on its own through a channel, what if there is a carrier that has a binding site for sodium and glucose? And as soon as sodium and glucose both bind, then it will move both of them in simultaneously. So it's like you were on your way to Florida and somebody said, well, I want to go to Florida, but I'm not going to pay for gas and I'm not going to drive, right? The answer would be no, unless it was glucose, right? So unless it was the hottest girl imaginable and you were 22 and still willing to put up with hot people's crap, right? Okay, so remember that time when you used to be willing to deal with that and now you're like, oh my God, I'm sure somebody hates him. I know somebody hates him. So what happens is sodium is motivated to go inside the cell anyway. Sodium has an electrochemical gradient such that it will move inside the cell very readily, and I've been working my butt off to get it to move inside the cell really readily. And instead of bringing sodium in through a channel, I'm going to have a binding site for sodium, a binding site for glucose, and then bring both of them in. Does that make sense? Do you see that that is cheaper than pumping glucose in? And it allows you to completely ignore glucose's concentration gradient because I am not using ATP, right? And I don't care whether there was already glucose in there, I can still bring it in this way, okay? But do you see that after you bring in a few glucoses, it's going to disturb your concentration gradient for sodium, is it not? Sodium, sodium, sodium. Yes? But is it cheaper? Well, first, how are you going to fix the concentration gradient that you just screwed up? Yeah, you're going to pump it back out. Yeah? Okay. But now I want you to compare. If I were pumping each glucose in, and let's just say it took an ATP per glucose, right? Because glucose is relatively large. Not a whole bunch of them fit into a carrier. So it took 1 ATP per glucose. Can you see that even if you eventually had to pump out the sodium for bringing the glucose in, it would be cheaper? Because how many glucoses would you get in before you had to use an ATP to fix homeostasis? Think about your sodium-potassium pump. How many glucoses can you get in before you have to use an ATP to fix it? If each time a sodium comes in, a glucose comes in. How many glucoses can you get in before you have to use an ATP to fix your sodium concentration gradient? 3. 3 sodiums out for every time you use the pump. So do you see? I get 1, 2, 3 in before I have to use ATP for the pump. Yes? Do you see how that's cheaper than pumping every single glucose in? Do you see? Okay. So you don't? If I used an ATP for each time I use this, right? So if I had a glucose transport mechanism and I wasn't using sodium as the driving mechanism, I was using ATP, right? Then every single time I brought glucose in, I would use an ATP. And that means for 3 glucoses, I would use 3 ATPs, right? Does that make sense? Not for 3 glucoses, I would end up using 1 ATP. Because what's going to happen after this is after you use this sodium glucose transport carrier, right? You get to use it 3 times before you actually have to fix. And the way that you're going to fix it is with the sodium-potassium pump. So what you are going to do, oops, not that one. What you are going to do is there are 2 steps. Okay, 2 steps of secondary active transport. The first one doesn't require ATP, but the second one does. Yes? The first one was what? What is this transport mechanism? It's a carrier. And if you diffuse through a carrier, what is it called? Facilitated diffusion. This is simply facilitated diffusion with the hitchhiker, right? Still facilitated diffusion, yeah? You're diffusing something through a carrier. You're just bringing something along with you, okay, without paying for it, okay? So this is facilitated diffusion. So the first step is facilitated diffusion, and then the second step is going to be active transport. So it is, first step, a carrier protein moves 2 substances at the same time. Let's say they go in the same direction, but they don't have to. The first substance, sodium for instance, moves into the cell down its electrochemical gradient, which I just worked very hard to make sure was going to happen. Okay. The second substance, something valuable that you don't always want to pay for, right? glucose, or an amino acid, tags along because the carrier has a binding site for both substances, and it doesn't actually carry until both substances are bound, right? So I've got sodium sitting in the driver's seat, but it's not going to go anywhere until glucose gets in the passenger seat, yeah? And then when they're both there, then sodium's concentration gradient will actually allow for the carrier to move, okay? So if two substances are moving the same direction, it's called symport or co-transport because you're moving them together. But just so you know, you can actually use sodium to pop something out of the cell too, like a revolving door. So I'm going to show you an animation that shows you. So ultimately what you're trying to do is to bring something desirable into the cell without expenditure of ATP directly. Yeah? Okay, so let me show you an animation. So this is sodium-potassium. I'm sorry, this is the co-transport. So what I'm doing is, the thing I don't like about this animation is that they don't show you the binding sites, because this has shape-specific binding sites for sodium and the sugar, the glucose. Okay, and then what's going to happen is you get to bring some glucoses in, but then eventually you're going to have to fix the disturbance in the sodium concentration that you made, right? So you're going to have to use the sodium-potassium pump. So it's two steps. First step, facilitated diffusion with the hitchhiker. Second step, active transport. Okay, so which one is ultimately cheaper? Secondary active transport or primary active transport? Primary. No, secondary is cheaper. Yeah, secondary would be a cheaper mechanism. Primary would be, for instance, pumping glucose directly into the cell. Does that make sense to you? Now, you can, by the way, make things go the opposite direction. A lot of people don't like this concept, but if you use sodium's concentration or electrochemical gradient, you can also pop something out of the cell. So let's just show you right here. So this one is using sodium's concentration gradient to pop calcium out of the cell. You can pump calcium out of the cell too, but this is cheaper. Yeah, because you're going to get more calcium moved for less ATP. Okay, so this is called secondary active transport, and it is a two-step process. So sodium, when do you use this? Sodium can be used to bring glucose inside the cell without paying for it directly. It can be used to bring amino acids inside the cell without paying for them directly. You do eventually pay, but you only pay to replace homeostasis. It is also, sodium is also used to get H+ out of the kidney. So you'll run into it later this semester too. Okay, so there is a summary for those of you who like, I like this kind of organization. My brain just works this way. And the reason I think it's useful is because sometimes you just look at my notes and you don't actually do comparison contrast to figure out how, for instance, simple diffusion versus diffusion through a channel versus diffusion through a carrier versus primary and secondary active transport, how are those different from one another? Do they use energy? Do they use a protein? If so, is it a channel or a carrier, right? If they use energy, are they using ATP energy or are they using an electrochemical gradient to drive it? And this actually allows you to do comparison contrast so that you figure out whether you got it. It's not something that you want to do in class. You want to do it at home. Figure out whether you can actually separate when you would use one versus the other. Okay, go ahead. Huh? It's in your textbook. This is in your textbook. It's table 4.2 right there. Okay, so the last type of transport in chapter 4 is endo and exocytosis. Those are broadly called vesicular transport because you move things in vesicles, yeah? And they are the slowest and the most expensive of all the transport mechanisms that we have learned. So endo and exocytosis, let's talk about those. So when you do, so if you're doing vesicular transport, what happens is you have something that's a membrane-bound bubble and it's got a phospholipid membrane around it. And then you are moving the vesicle against the plasma membrane, or you're moving something in via a vesicle. And like I said, we actually do not know how ATP is used in these transport mechanisms, but we know that they won't happen in the absence of relatively large amounts of ATP. So these are really energetically expensive. And we wouldn't do them if any of the other ones were possible. So what kinds of things do you tend to move via endo and exocytosis? Sugars and amino acids can move through carriers. Huge things like big proteins, multiple amino acids stuck together, maybe hundreds of amino acids stuck together, right? You can't get it through any other mechanism. So this is exocytosis, which is moving things out via vesicles, and then we'll talk about endocytosis. Exocytosis, let's say this was a pancreatic cell, and I made insulin, okay? And I made insulin in the pancreatic cell, and then I packaged it up in the Golgi apparatus, right? And in the Golgi apparatus, I packaged it up in a vesicle, right? And then insulin doesn't do anybody any good unless it gets into the bloodstream, right? So what I have to do is get it out of that pancreatic cell, right? And so the way that you do that is just smash it up against the plasma membrane, and then you depend on the fluid mosaic nature of the plasma membrane so that the phospholipids will sort of join with one another. And then you end up dumping the insulin and keeping the phospholipids. Yes? Right? Slow, energetically expensive. That's exocytosis. Usually large things or things that have serious solubility problems, but usually very large things, like big proteins. Okay, so with endocytosis, there's actually 3 different kinds of endocytosis depending on what you're bringing in and how. If you're doing phagocytosis, you're bringing in big solid chunks of something, like a white blood cell taking in an E. coli, right? If you're doing pinocytosis, pino means to drink, so you're taking in something that is dissolved in the extracellular fluid. And then the last type is the slowest type, and we'll talk about that. So phagocytosis, you probably ran into, like, hold on, here's phagocytosis. Where are you? Phagocytosis is like this one. Oh, balls. This one. So phagocytosis, so I want you to imagine that this is my cell membrane, yeah? And this is a bacteria, some kind of coccus, because I'm round. And what's going to happen is it is going to reach up and fold around the bacterium. White blood cells, macrophages are famous for doing this. And then as it folds, you again depend on the fluid mosaic nature so that it actually seals up. And then you end up bringing a chunk into the cell. And if it was some kind of pathogen, then you just use it with a lysosome and whoop up on it or whatever. Does that make sense? So that's phagocytosis. Pinocytosis is when you just fold around the extracellular fluid which has something dissolved in it, and you bring in a bubble of fluid, but it's dissolved, it's not in big chunks, right? So that's pinocytosis. And then the slowest kind is called receptor-mediated endocytosis. And receptor-mediated endocytosis is when I'm trying to bring something very specific inside the cell that won't move by any of the other mechanisms. Otherwise, I wouldn't be using this one. And so what you do is you have to wait until this thing that I'm trying to bring in, which is this purple triangle, binds to receptors. And once it binds to receptors, enough of them, then it will trigger endocytosis. So it's receptor-mediated endocytosis, right? Why would you generally not do endocytosis when you got one single molecule bound? Too costly, yes, very, very expensive. So what happens is you wait until multiple ones are bound. This is the mechanism for transporting some forms of really insoluble iron, and there's also a form of cholesterol which is not very lipid soluble and hard to transport. So you wait until it's bound, and then you do endocytosis, and then you end up with a bubble with, yes, extracellular fluid and other unintentional stuff in it, but importantly, it's got the stuff that you really wanted. Can you see that this would be the slowest of all the endocytosis mechanisms? So if you're really going, cheapest, fastest way first, this is super duper slow and really expensive, right? So it's definitely not plan A, right? Does that make sense? Okay, so that's called receptor-mediated endocytosis. Okay, so now to apply all of this, what I want to see is if you guys can quiz yourself by looking at each of these transport situations and telling me what kind of transport you would do. What is the type of transport? We're trying to get something across the cell membrane. Does it use a channel, a carrier, or pump, right, which is a carrier plus ATP? And then, of course, is energy required, right? So in each of these situations, that's what I want you to do. So how would each of these substances cross the cell membrane, and why, importantly? What type of transport protein, if any, is necessary? So oxygen going down its concentration gradient. What type of transport? What? Simple diffusion. And what is it about oxygen that allows you to do simple diffusion across the cell membrane? It is lipid soluble. Yes, it is lipid soluble. Very good. Okay. Potassium down its concentration gradient. What? Any kind of channel really, but leak or gated if the gated one's open. Now, my question to you, defend that by telling me why you couldn't do simple diffusion of potassium down its concentration gradient. Exactly. Good. Okay. An amino acid moving into a cell for protein synthesis. Facilitated diffusion. Yes, I agree. If the concentration didn't favor it, you could also use secondary active transport. So facilitated diffusion, if the concentration gradient favors it, yeah? The concentration gradient doesn't, then you'd probably use secondary active transport. Does that make sense to you guys? Okay. A large antibody protein secreted by a white blood cell. What? Not endo. Exocytosis, yes. Exocytosis is moving it out. Does that make sense? Now, back up. Tell me why you couldn't use channel or a carrier. Too big, exactly. Too big and not lipid soluble, correct? Okay. Water moving from a cell's ICF into a highly concentrated saline solution. So it's moving out of the cell. What type of transport and any proteins required? It's osmosis. Does it require any transport proteins? No, it doesn't require any transport protein. I mean, unless you consider the legendary aquaporins, which I don't usually ask you about because they never even draw them in. But okay, calcium being pumped into the calcium-rich ECF to allow for muscle relaxation. What? Go ahead. I know, but now tell me why you couldn't get it to move via a channel or a carrier. You're going against the concentration gradient. And so regardless of whether it was large or small, if you're going against the concentration gradient, you still need to use, what is a pump? A carrier or a channel? Pump is a carrier. It has to have a shape-specific binding site plus ATP equals a carrier. So do you see this example? So I want you to know that I'm not asking you to memorize these. I'm asking you to be able to look at a situation, and as long as I'm clear enough about the way I write the question, then you go through in your head, and if you don't use one of the cheap, fast ways, you need to know why you're not using it, right? So then you go on to the next one. Does that make sense to you guys? Okay, so that's good practice for you guys. Okay, so now what we're going to do is we're going to take all of that crap and we're going to put it into the context of the human body, which you probably didn't do in 1610. And basically, instead of just trying to get something across a single layer of the cell membrane, we're going to try to get it all the way across an epithelium. Because that's how you get things into the body or out of the body, correct? Right? Okay, so a great example of what we call epithelial transport or transepithelial transport is trying to get any nutrient or water or ion from the lumen of your digestive tract all the way across the intestinal epithelium into the bloodstream, right? And so before you get into that, you have to remind yourself that you need to remember the basic anatomy of an epithelium, right? So if you're looking at an epithelium, and let's say this was the epithelium that was lining your small intestine where you do the vast majority of your absorption, right? Then, of course, you would have the lumen. And what's this surface of the epithelium called? Apical or apical, yes. That makes sense? The apical surface of the epithelium. Notice the microvilli, which does what to the transport rate? Increases the transport rate. So you're going to have to move from the lumen across the apical surface through the intracellular fluid, yes? Across the basal surface, also called basolateral, through the interstitial fluid into the bloodstream, okay? So there's going to be a transport mechanism here, yes? And then a transport mechanism here. Potentially a transport mechanism here, but you don't usually have to worry about it because those capillaries are fenestrated. So if you get them close, they'll just go straight in to the capillaries. So you just have to worry about those two. So what we're going to do is we're going to look at specific instances of how you get things across, and you're going to use those ideas of cheapest, fastest first. And we're going to try to do transepithelial transport all the way through and into the bloodstream of 3 different things. We're going to do sodium, we're going to do glucose, and we're going to do water. Okay? So if we do transepithelial transport of sodium, what I want to talk about is I just ate Cheetos, and I want that sodium to end up in my blood plasma because, of course, I'm going to need it, right? Okay? So what I want to know is how am I going to move sodium across the apical surface? So right here, what kind of protein, what kind of transport, and why? So it doesn't tell you because you should be able to figure it out. No, it's not going to be active transport. What? It's a channel, okay? And it doesn't really matter whether it's leaked or a leak or gated, but it's a channel. What is it that you know about the intracellular concentration of sodium, which will allow you to move this through a channel instead of having to pump it? Intracellular concentration of sodium. Yes. Don't you work your little butt off to make sure that the intracellular concentration of sodium is low? In addition, what is also the intracellular, the charge on the inside of the cell membrane? Negatively charged, right? So do you see that this can be a channel? Okay. Now, one last question, back up. Why can't you diffuse it straight through the plasma membrane? Charge, exactly, right? That makes sense? So everybody understand that this is a channel? You got it? Okay, so now I still haven't accomplished what I want. I want it in the bloodstream, right? So I'm trying to make it from my Cheeto across my intestinal epithelium into my bloodstream. So now my question for you is, how do I get sodium out through the basal or basolateral surface? Sodium-Potassium pump, but defend it. Why do you have to pump it here when you didn't have to pump it there? It's going against its concentration gradient, right? Didn't we say that sodium was low here and high here? Yeah? So now you have to pump it out, right? Does that make sense? Okay, got it? So you have to pump it out into the extracellular fluid, and then as soon as you get it close to the fenestrated capillaries, then it gets carried away, right? Okay, what about potassium? Well, potassium gets pumped in at the same time, but then it diffuses straight back out the leak channels because there's boatloads of potassium leak channels, so you don't really have to worry about it. Potassium just does what it does, okay? Are we okay with that? Do you understand the concept? Do you see that, like, if you really get this, this isn't memorization. It's logic, isn't it? What do I know about concentrations, right? And how can I put that into this concept, right? So now, so if we look at the summary, okay, so the transepithelial transport of sodium. So the summary is sodium comes into the epithelial cell by diffusion through a channel, leaves the epithelial cell by primary active transport, and then moves passively into the blood plasma. Does that make sense? Okay. All right, so let's try another one. Now I'm going to do transepithelial transport of glucose. I just ate a Snickers bar. Yes? And a Snickers bar, like any other sweet worth its weight, has salt and sugar in it. Does it not? Okay. Have you ever like forgotten to put salt in your chocolate chip cookies? And they taste like butt, don't they? They taste terrible. You have to have a little salt. Okay, so it's going to have some salt and it's going to have some glucose. And so what I'm going to do is I'm going to do transepithelial transport of glucose. I'm going to look at this figure right here. Okay, now the thing I want you to understand is that I ate a Snickers bar and I have been absorbing some glucose, and in all likelihood the intracellular concentration of glucose is already relatively high. Okay. So I could pay to bring glucose in, yes, because I still want it, right? Because I don't want to poop out the most valuable nutrient that I could possibly have. I want to get all of it in if I can. So now I'm going to try to get glucose in regardless of glucose's concentration gradient. And how am I going to do it? Secondary active transport. And what kind of protein is that? Right there. What kind of protein? Channel carrier pump. Channel carrier pump. What is it? Yeah, it's a carrier. It has a shape-specific binding site for both glucose and sodium, but it does not use ATP, right? Okay, so this is a carrier, and as soon as glucose and sodium are both bound, it's going to bring them in together, yes? But we haven't accomplished our goal, which is ultimately to get sodium all the way into, glucose all the way into the bloodstream. So now we have to think about, okay, we got glucose in. How do we get glucose out? Okay, so how do we get glucose out? I'll tell you something to make it a little easier for you. Once you get glucose out, it doesn't hang around. You dump it directly into the bloodstream and carry it off. Okay, so now how do you get glucose out? Think about it.
Primary active.
Nope. Can't do simple diffusion because of what about glucose? You need a carrier, don't you? Can't do simple because of solubility and size. You can't do channel because of, why can't you do a channel for glucose? Size. Size. You can't do channel because of size. Carrier is your next thing. And then you always, all you have to figure out is, do I need ATP or not? Well, I just told you we've been carrying away the glucose, yeah? High here, low here. We can use a, what is that again? Carrier. And what is the transport mechanism called right there? Just right there. Nope. It's facilitated diffusion. It's just diffusion through a carrier. Yeah? Okay. All right. And then once I get it close to the fenestrated capillaries, it goes straight in. Okay. But then, of course, I have to deal with the fact that if I bring enough glucose in via this mechanism, I'm going to disturb the concentration gradient for sodium. So how do I take care of that? I have to pump it back out. Yes? Pump it back out. And you told me last time, but I want somebody else to tell me this time. Why do I have to pump sodium out when I could pull it in via diffusion? Concentration gradient, right? I have to pump it out. So you pump sodium out using the sodium-potassium pump. Is that okay? Okay. So are you starting to get it? So with each thing, what you do is you don't look at it and wait for the figure to tell you. You should be able to predict and then confirm it with the figure. Okay? So why would I move it here? What's the transport mechanism? What kind of protein do I need and why? Okay, so do you see, can you hear by the way that I'm talking that if you just memorize it, you probably will not get my questions right? Because I am not going to ask you necessarily the one that you memorized. I'm going to throw you a situation, give you enough information about it that you should be able to figure it out in context, right? So maybe next time it won't be glucose, but it'll be an amino acid, right? And the amino acid would also be relatively, it's mid-sized and water soluble, right? Okay, so this mechanism right here is basically secondary active transport, right? Okay, so that's transepithelial transport of glucose, and I have along my intestinal epithelium carriers that have binding site for sodium and glucose and move them together. I also have carriers that have binding sites for sodium and amino acids, each one of the amino acids, and move them. It's the cheapest way I can get them in if I've already got them in and I still want them, right? So obviously you want as much of those nutrients that are incredibly valuable as you can to be absorbed, and you don't want to have to pay to get them in every single time, right? So you're going to use secondary active transport. Okay, so the last one is transepithelial transport of water. So this question asked, okay, I ate a Cheeto, I ate a Snickers bar, and I'm going to drink some water. And how do I get the water to move from the lumen of my intestine into my bloodstream? Okay, so how do I get it to move? Well, your textbook has this crappy picture. Like that picture has anything in it. Look at that picture. Does that tell you anything? It's just a bunch of balls. Does it look like confetti? Doesn't tell you anything. So that pointless picture is not very useful to me, so I'm going to use this one. And I want you to think about the story that I'm telling and see if you can figure out what it means. So if I just look at sodium for just a second, just to simplify this. Do you see what sodium is doing is moving from the lumen through the ICF to the interstitial fluid and then to the blood plasma? Do you see that? Large concentrations of sodium. The same thing I'm doing here, only I'm moving glucose simultaneously. Now, what I want you to see is as sodium's concentration moves from here to here to here to here, what is water's concentration doing as sodium's concentration is increasing in each place? It's decreasing, right, relatively? So do you see what you're doing? As sodium's concentration increases inside the cell, then water's concentration decreases. And as sodium's concentration increases in the interstitial fluid, then water's concentration decreases, and same in the blood plasma. So what I'm doing is I'm creating an osmotic pull for water to move into the bloodstream. And that's how your water moves into your bloodstream. And what transport mechanism is that? It's osmosis, yes, it's osmosis, right? And does it have to be sodium? No. All solutes suck, but sodium sucks the most. And dependably, we are Americans. We've got plenty of sodium in our diets, right? nobody's like poor on sodium concentration, although it will happen with these solutes as well. As long as the solutes can't diffuse straight back out, then they're going to create an osmotic drive for water to move in. Does that make sense? So that's just osmosis is what it is. So that's primarily how you actually end up getting water into your system. And you've probably experienced this. If you eat the whole damn 1 pound bag of Cheetos, how much water are you actually going to absorb and retain the next day? You're gonna be all, can't get your rings off, somebody shrunk your shoes, your pants went down a size somehow. Yeah, and it has to do with the amount of solutes that you're taking in, and then the water will actually sort of be retained as well. Is that okay? Okay, so with that in mind, what you're going to have to do on your test, which is a week from Wednesday, is you're going to have to do transepithelial transport of either sodium, glucose, or water, and I'm not going to tell you which one because then you would only study one, right? And you're going to have to lead me through it, okay? And you can do it in an image if you want to, but it has to be really well labeled because I'm not a visual learner, right? So if you draw me an image that looks like this, I'm just going to go, I'm moving on because I don't even know what the hell you're doing, right?
Something like that, though. Just
Like this one? As long as you label them.
Like that.
Yeah. You got to label them. You got to tell me that's a channel and that's a pump, right? You have to tell me what they are. And I'll make sure that I write the question to ask you what kind of transport protein it is and all that kind of stuff. Okay? So, all right. So, the last thing on this set of notes is simply filtration. They don't actually cover it in this chapter of your textbook, which I think is weird because, so we just talked about transepithelial transport, which is moving something across an epithelium. And all of the examples that we gave were in the intestinal epithelium. But isn't filtration just moving things across an epithelium as well? It's just like you use pressure as a driving force instead of worrying about concentrations. So I'm going to talk about filtration here. Your textbook doesn't talk about it at all until it gets to the kidney, the urinary system, which is at the very end of the semester. But filtration in my mind goes here. It's a good place to talk about this transport mechanism. Filtration is movement of water and small solutes across a membrane, but it doesn't give a crap about concentration gradients. It's exclusively pressure driven. So, you know, fluids, all fluids move from high pressure to low pressure, right? So if you turn on your tap, the reason the water comes out is because it's lower pressure out here than it is in the tap, right? And the pipes behind. So filtration will move things, water and small solutes, across a membrane from areas of high pressure to low pressure. And the metaphor that works, it worked way better in Texas than it does here, is making coffee in a coffee filter. When I first got here, people are like, huh, what? Like, you should see it on TV, right? I had to teach my mother-in-law. My mother-in-law is of the predominant faith, and we had European guests, and she's like, well, I have to offer coffee. I was like, I've been telling you that for years. But she's like, how does this work? So here's how it works, Johanna. The membrane has pores, yeah? And so it's a coffee filter, right? And so what you are going to do is you are going to put coffee in the coffee filter, and then you're going to put water in. It's going to pull things through based on size and water pressure, right? So water's going to pull it through. And it doesn't require energy, right? Because you would make coffee, even if you didn't plug the thing in, it would just suck. That's all. It would be terrible, terrible coffee. So you are actually going to pull it through. But it's also not selective about what it's moving across, because if you put dirt and coffee in the coffee filter, you'll pull all of the small pieces of both, right? Dirt and coffee and red Kool-Aid, and you'll pull all the small pieces of both because it's driven by pressure and it's only selective by size. So it's going to pull anything across that will fit through the holes. So you can keep all of the large pieces behind and let all of the small pieces come through, unless, of course, you increase the pressure dramatically, because if you were making coffee and you hooked up a fire hose behind the coffee filter, you could put a bowling ball through as long as you never wanted to use the membrane again, correct? Okay, so when does this exist in the body? Everybody knows that it exists in the kidney. But let's broaden it, because it exists in every single capillary in your body, because what I have in capillaries is two fluids surrounded by the capillary membrane. It's an epithelial membrane, right? And I've got the blood and the blood plasma on this side, and I've got the interstitial fluid on this side. But one of those fluids is dependably higher pressure than the other one. Which one? Blood plasma or interstitial fluid? Yeah, blood plasma. Which one has a pump hooked up to it? The blood, right? So what happens is you've got capillaries and they've got fenestrations or sometimes even huge holes. And what I have is blood plasma in here, interstitial fluid in here. And what happens is I push fluid. That's where the interstitial fluid comes from, ultimately, and how it gets put back in. So it gets pushed out, but not just water. All of the small solutes, all of the ions and the little things that fit through the holes get pushed out that way as well. Okay? Happens at every single capillary in your body. All of the capillaries in your body, except for your kidneys, combined, filter out about a gallon of solution, water and the small solutes, per day. But what they shouldn't do is filter out whole blood cells, right? It's a bowling ball. Or big clotting proteins, right? Those should end up staying behind. So let's talk about the kidneys, and then we'll talk about hypertension and what can happen with hypertension, which is chronic high blood pressure. So the kidneys are just set up for filtration better than the rest of the capillaries in the body. They're set up for filtration better because there's slightly higher pressure, which would of course increase filtration, right? Way more surface area, yeah, which would also increase filtration. And then they're really porous or holy, not the kind, but the other kind, right? They're really holy, right? And so how much better are they than the other capillaries? The kidneys combined filter about 47 gallons per day. The rest of the blood vessels, or the rest of the capillaries collectively, 1 gallon. So the kidneys are like badasses of filtration. So that's what the kidneys are going to do, and they filter out, and primarily what they're doing is a lot of those small solutes are waste products like urea and uric acid that should get pushed out via filtration. Does that make sense? That's how it ends up in your pee. That's a large part of how it ends up in your pee. Are we okay with that? Okay, so 2 questions. Chronic hypertension, untreated high blood pressure. Can you see conceptually how that might lead to kidney failure? Yeah? It's hooking up a fire hose behind your coffee filter, yes? And it can completely trash your membranes, and you can end up with first, large proteins in your urine, and second, whole blood cells in your urine, and then third, your kidneys are just not filtering for you at all. Just like, sorry, I don't know what to do. I don't really have a membrane to push things across, right? So you can end up with kidney failure associated with that. So let's talk about one of the things that you could do with kidney failure regardless of why it was caused. So if your kidneys can no longer filter out like nitrogenous waste products like urea and uric acid and ammonia, those are waste products primarily from protein and nucleic acid breakdown. And they've become problematic if they increase in concentration in your bloodstream because they will screw up your pH, among other things. Can you see that, for instance, if you had kidney failure, your urea concentration in your bloodstream could start going up? Does that make sense? Let's just concentrate on urea, okay? There are other things that you're going to be moving as well, but if you concentrate on urea, one of the things that you can do if filtration fails and your kidneys are no longer cleansing your blood like they should via filtration is you can use dialysis as a substitute for cleansing your blood. So dialysis is largely, and this is hemodialysis, there's several different kinds. This kind is going to cleanse your blood. So what I'm going to do is I'm going to take my blood out of a vessel and put it back in, sometimes to a different vessel, sometimes the same one. And then as my blood, I'm pulling it out, I'm going to run it through a dialysis machine. And inside the dialysis machine is going to be a dialysis chamber. So inside the dialysis chamber, and I brought one for you guys, is going to be something that should make sense to you. So what I'm going to have is I'm going to have 2 fluids, and I'm going to have a really super duper curved membrane between them. It's called a dialysis membrane. And in one of them, I'm going to be putting, blood is going to come in and blood is going to go out, okay? And this is going to go back to the body, okay? And because my kidneys are failing, what can you tell me about my relative concentration of urea? Should it be high or low in the bloodstream? It's high, yeah, otherwise I wouldn't be doing this, right? Okay, and then in this one, I'm going to be putting in dialysis solution. Oh wait, the other way. I do countercurrent because you actually get better transport that way. So dialysis solution goes in that way and out that way. So dialysis solution. And then what happens when you get it out this side? You don't circulate it, you just dump it, right? That makes sense? No reason to reuse the dialysis solution. I put in clean dialysis solution, I dump it when it's dirty. So dump it. And in order to make this cleanse the urea out of your bloodstream, what can you tell me about what the concentration of urea should be over here? Low. It's actually 0 urea concentration, like none in there. And then if you want urea to move from the blood to the dialysis solution, you better make this membrane permeable to what? What does it have to be permeable for the urea to move? Yeah, it has to be a urea permeable, right? If you designed a dialysis membrane that wasn't urea permeable, you would do a really expensive, pointless activity. Now, if someone is going through kidney failure and they're doing hemodialysis, depending on how badly their kidneys are failing, they could have to do it for an hour per day or 8 hours per day or 12 hours per day, depending on how badly their kidneys are failing, because you really start to build up this urea concentration. So what you have here is, I told you the membrane was curvy, but the membrane is actually so curvy that you can't even tell it's a membrane. And so what you have here is the dialysis solution comes in this side and goes out that side, and the blood comes in here and goes out here, right? And if you see, you can see that the membrane is just outrageously large surface area. You guys can pass that around if you want to. So what else should you make sure is true about the dialysis solution compared to the blood? Tonicity. Yeah, it should be isotonic, otherwise you're going to blow up your blood cells or shrivel them up, right? So you want to make it isotonic and you want to make it really low in concentration to whatever toxins you're trying to remove, right? And then you have to design the dialysis membrane so that you know that it actually is permeable to whatever toxins you want to move. And it shouldn't be permeable to things you want to keep, like glucose, right? That makes sense? So this is called hemodialysis, and that's what you do. So, but hemodialysis, so cleansing urea out of your bloodstream in your kidneys is actually based on filtration, but this isn't based on filtration. What is it? What's it based on? It's just diffusion, right? It's actually simple diffusion because you made it permeable, right? So that's based on diffusion. That makes sense? Okay, so that's hemodialysis. There are a couple of different kinds of dialysis that I don't actually know quite as well. One of them is peritoneal dialysis, in which they do your fluid in your peritoneal cavity instead of your blood. But hemodialysis is the one that's been around for a while. I don't know what situations they use one versus the other. Okay, is everybody okay with that? Okay, so now this next set of notes is where we are going to start diverging from 1610, and we probably won't really ever go back to have much overlap with 1610. And so what we are working on with all of this business is trying to get here, trying to get to the point that we can start understanding the concept of membrane potentials and action potentials. This is chapter 7 in your textbook. And so in order to get here, we're going to just acknowledge the things that we've said before, and we are going to look at how we got here, and then we're going to start changing it. But in order to do that, let's to make sure that everybody kind of understands how we got here. So things that you know, you should know these. I'm just going to say them in a slightly different way than I did before. The ion composition of the ICF. Actually, you don't know quite all of them. The ICF has a high concentration of potassium, 30:1, low concentration of sodium, relatively, right? And although I didn't ever tell you before, there are, of course, negatives in here, right? What's the primary source of negatives on the inside of the cell? They're big, huge, impermeable, negatively charged proteins. Because obviously the cell has lots of proteins, right? Structural proteins and enzymes and all of that. And proteins tend to have a slight negative charge. And they're trapped, right? Because they're huge and you don't move them on accident. So they pretty much just stay in there, right? Is that okay? So the primary source of negative charges, meaning these negatives that are revealed, are mostly because of the proteins on the inside, okay? And then if we look at the extracellular fluid, The ion composition of the extracellular fluid has a high concentration of sodium, a relatively lower concentration of potassium, and then although I didn't tell you this before, the extracellular fluid primary source of negative charges is chloride ions. Not exclusively, they're just in really high concentration in the extracellular fluid. All right, so phospholipid bilayer exists between them. That's how we can maintain the concentration gradients. And of course, now we know that we have set up a situation that ions can and will move down their electrical gradients or their chemical concentration gradients. They can't move directly through the lipid portion of the cell membrane, but they can move through a channel or even a carrier if they needed to, okay? So what we established here, although we didn't call it this is called resting membrane potential. But let's acknowledge that this thing is really never going to be at rest, is it? Because what would happen if the cell completely rested? For instance, your sodium-potassium pump. You would first lose your concentration gradients, yes? You'd lose your electrical gradients after that, right? And then the cell would no longer have homeostasis, and if you left it there for a while, the cell would no longer be functional. So this is called resting membrane potential, because this is the least that the cell ever does, even though it's not restful. So we already sort of set up how this came to be. So I'm going to explain it one more time, and this is your opportunity to make sure that you get it, and then we're going to start changing it. So it's super duper important that you understand the story of how it gets here. So one more time. Before the charge existed, all we had to deal with was concentration gradients, right? So we had concentrations, no charges yet, and we put in our leak channels. And initially when you put in your leak channels and there is no charge to deal with, then which ion initially diffuses faster? Potassium diffuses faster out than sodium diffuses in. If you let that happen for just a second, right, then what charge accumulates around the outside of the cell? Positive. And what charge is revealed on the inside of the cell? Negative. Good. Okay. And then after that occurs, which ion is going to speed up? Sodium is going to speed up because now it's moving toward an opposite charge, right? And potassium is going to slow down, right? So does everybody understand how you got to be charged? Yeah? Okay. So if you understand how you got to be charged, then you can start to conceptualize how we can change it. So this is called resting membrane potential. So one question before we leave the resting membrane potential and start talking about numbers is, what about these negatives? Why do we not even acknowledge them, really? Why do they not really affect the charge? There's a couple of different reasons. First of all, the proteins are impermeable. They're really trapped, right? So they don't really wiggle in and out of the cell, so you can kind of ignore those because they're just going to stay in there. But chloride is not huge. Chloride has a high concentration outside, and so it wants to diffuse in. But it actually wants to diffuse out via its electrical gradient. And it diffuses in, down its concentration gradient, and out, down its electrical gradient at about the same rate. So you can kind of ignore it, because it's like water in an isotonic solution, right? Where does it move? In and out, but no net movement. Does that make sense? Okay. So that's why you get to actually ignore something for a change in physiology, which hardly ever happens. Okay, so this is a cell at rest, and when the cell is at rest, you call it steady state or resting membrane potential, and your textbook has this figure. We don't look at it in class, it's too complicated, but what I want to tell you is this figure, which is 7-8, reviews for you exactly what we just did, right? We start out with no channels and just concentration. We add in the channels, we let them move, we get a charge, right? And then we balance it out with the sodium-potassium pump. So it has that, if you want to read through it that way. And then also in interactive physiology, where is it? Interactive physiology has an animation that will build the entire thing for you. And you can play with it, and you can open things and close things and stop the pump and start the pump. It's pretty helpful, especially for those of you who are very visual or kinesthetic learners. So it's probably a good way to sort of review and make sure you've got it. Okay, so now So what I'm going to say now about the cell is that the cell membrane, now we describe the cell membrane as polarized. Do you see that it's positive on one side and negative on the other? And it's polarized in such a way that you can measure it with a voltmeter, little bitty teeny electrode that they actually stick in, okay? And you measure it with a voltmeter and it comes out to be a number, but let me tell you what the number is actually trying to tell you. The cell membrane is polarized, and when you measure it with a little electrode, it actually hits the extracellular fluid first, right? And uses that solution as the standard, yes? And then hits this one second and compares that one to the standard. So it ends up as a negative number, because that is negative with reference to that. Does that make sense? And it measures at about minus 70 millivolts, which won't be very meaningful until we start to change it, and then you'll kind of understand what the 70 has to do with. So that's how different the inside and the outside are. It's how polarized the cell membrane is at rest. Does that make sense? Okay, so it's minus 70 millivolts. Now that's just an average, because neurons are slightly different than muscle cells, which are slightly different than other cells, but minus 70 is an average, and some of them are 60 and some of them are 80, okay? So minus 70 millivolts, so cells are polarized at rest. So now what I'm going to tell you is you're going to know several different things that are homeostatic for the cell. Okay, this is homeostatic for the cell. Okay, this is homeostatic for the cell. This is homeostatic for the cell, and that number is homeostatic for the cell. I mess with it all the time, but I have to put it back. Okay, so if 120 over 80 is the blood pressure that I want, then why do I ever get it any different than 120 over 80? Because sometimes I have to accomplish something that requires it to be different than 120 over 80, right? I have to do some crap, okay? So you always have to put it back there. So minus 70 millivolts is what we call resting membrane potential. That is resting membrane potential. This can change rapidly, but it must be returned to maintain homeostasis. So the next thing that I want to talk about is if I have this electrical setup, and so I've been dealing with only leak channels to this point. Do you guys agree? Only leak channels? But now I want to deal with gated ion channels, which also exist in the cell membrane. So if I open a gated ion channel, can you see that all of this work that I put in means that ions will be motivated to move across the membrane, either down their concentration gradient or down their electrical gradient, right? Okay. So the two forces that I have set up to motivate ions to move is called the electrochemical gradient. And the electrochemical gradient is responsible for the transmembrane potential. The electrochemical gradient is responsible for the potential for ions to move transmembrane across the membrane. Do you see the relationship of the terms? Okay. The electrochemical gradient that I just worked on setting up is responsible for the potential that ions will move transmembrane potential. Is that okay? Okay. So why do you care? Because when I move a charged ion, right? All ions are charged. Across a charged membrane, it creates a current. And that's how you get these electrically active tissues that we're talking about, okay? So when you learned about currents and electricity and physics, it was all kind of theoretical. You're trying to imagine what electron moves, how an electron moves across from one point to the other. But what I'm telling you is when you're dealing with electrical activity in cells, you've got a charged membrane and you move a charged ion across it and it creates a current, okay? And so now we're going to look at what kind of currents you're going to create. Okay, so this is resting membrane potential. We haven't changed it all. Every single cell in your body has a resting membrane potential that is similar to this. It's not identical, but it's similar. Now, neurons and muscle cells massively change this on purpose, and then you have to put it back. They massively change it either to send a signal or to trigger a contraction, and then you have to put it back. That means that these cells are going to be electrical because they're going to massively change the voltage on purpose by sending a whole bunch of ions across the cell membrane. It also means that these tissues are going to be energetically expensive because if I mess it up, what do I have to do? I have to put it back. Exactly, right? So you have probably figured out that nervous tissue, your brain for instance, is incredibly energetically expensive. It's a glucose hog, it's an oxygen hog, and one of the reasons is because it's starting from here and it's massively changing it in so many cells, and then you have to put it back. So what happens is this was baseline movement of sodium-potassium pumps. If you mess with it, you're going to have to use them dramatically more, and you're going to have more of them. Muscle cells also dramatically change this, and that's before they even contract. Okay, so see if this makes conceptual sense to you, and then we'll add some detail to it. So can you see how this number comes about? Even if you can't see why it's 70, can you see why it's negative? Okay, and it's a measurement of how polarized they are, how different they are. Does it make conceptual sense to you that that number would change somehow? You might not know how yet. If you really change the concentration of either of these, that number would change, right? Or either of these, that number would change. Does that make sense to you? Okay, and in a minute you'll be able to predict how it would change. What if you opened a whole bunch of gated ion channels that were not open? Can you see that that number would probably change if you opened a whole bunch of gated ion channels? Okay, depends on which direction the ion goes, but the number is definitely going to change somehow. Does that make sense to you? So this number is really easy to disturb. You can just move things, change the permeability of ions, or change the concentration of ions. What happens in your body all the time is that you change the permeability. Usually when you massively change the concentration, it's a pathological or pathophysiological condition. Like for instance, become depleted in potassium. But we'll be able to, by the time we finish this set of notes, understand what that would do to your nerve cells and your muscle cells. Okay, so this is polarized. This is at rest. Depolarized would mean the inside and the outside are becoming more electrically similar. Okay, you with me? Depolarized. And that number, if you were completely depolarized and they were not different at all, would read what? 0, yes? So if I depolarize, I move from -70 maybe to -30, yes? Maybe to -20 to 0. That's depolarizing. Does that make sense? Because not polarized is 0. You guys with me? If I hyperpolarize would mean the inside and the outside are becoming more different than one another, right? So what might the number do? Give me a number. -90, -100. Do you see that? That's further away from 0. So do you conceptualize what I'm talking about? Okay, so let me show you the graph that sort of shows you that. So here's a graph, okay? And this graph is showing you resting membrane potential, which is -70, and it's showing you depolarization, which goes towards 0. It can actually slightly overshoot 0 too. Hyperpolarization, which goes further away from 0, and then we'll talk about repolarization in just a second. So keep this in mind for a minute, and I'm going to give you a couple possibilities. My first question is, how can I accomplish depolarization? I'm going to give you two choices. I can either open gated sodium channels or open gated potassium channels. So I want you to sit there and chew on that for a minute and see if you can figure out which one would cause depolarization. Gated sodium channels opening or gated potassium channels opening. Anybody want to hazard a guess? Go ahead. Which one would cause depolarization? Opening gated sodium channels or gated potassium channels?
Would it be both? Because they're both going down the concentration gradient?
But which direction are they going?
One's going in, one's going out.
No, I'm not opening both of them at the same time. I'm opening one or the other.
Potassium goes out.
What do you think? Okay, let me tell you what's going to happen, okay? If I start from here and I open a gated potassium channel, for instance, I'm not saying that's right, then potassium will move out, right? And potassium is going to take positives from the already negative inside and put them into the already positive outside. Is that going to make the inside and the outside more similar, depolarized, or more different, hyperpolarized? Hyperpolarized, do you see? Okay, now I'm going to do the other thing. I'm going to open a gated sodium channel, and sodium's going to move in, right? It's going to take a positive from the positive outside and put it into the negative inside? Is that going to make the inside and the outside more similar, depolarized, or more different, hyperpolarized?
Depolarized.
Depolarized, more similar, right? Do you see? Okay. Do you see that opening a gated sodium channel would cause depolarization? Yes? Make the inside and the outside. Now, is there anything magical about sodium? Or could I pretty much use any positive ion channel that I know the ion would move in? Yeah. Right? It's nothing magical about sodium. So why is it true that we generally use sodium for depolarization? Because we just worked our butt off so that we know that sodium will move in when we open gated sodium channels. So although it's not a hard and fast rule, sodium depolarization, it's really common to use sodium for depolarization, right? Because we know sodium will move in, and it's going to move in fast too, right? Because the concentration gradient and the electrical gradient makes it move in really fast. So this, from here up, is most likely, but not always, a what channel?
Depolarization.
No, what kind of? Gated sodium channel. Yes, not leak. Talk about leak again in just a second. Not leak channel, okay? And then hyperpolarization. Hyperpolarization is making the inside and the outside more electrically different. Yes? And this is most often a what?
Potassium gated channel.
Gated potassium channel, exactly. That is a gated potassium channel. Does that make sense? Hyperpolarization? Okay, logic question for you. Now, I will tell you, and you'll figure out why next time, that it is really super duper common to do a massive depolarization because you open like hundreds of sodium channels simultaneously. And so you're going to depolarize all the way up to 0 and maybe even go slightly past. And depolarization happens really, really quickly. Well, I need repolarization to happen really quickly too. Repolarization will be getting me from here all the way back down to -70, okay? So I want to know what's a really, really quick way that I could get the number back to -70. Nope. So if I opened massive amounts of sodium channels and let sodium in to depolarize, how could I very quickly repolarize if I only cared about the charge? Open potassium channels, and let massive amounts of potassium out, right? Well, it's not hyperpolarization unless you start from resting. Okay, if you start from resting and go down, it's hyper. If you start from depolarized and go back to resting, it's repolarization, right? But it's the same concept. Do you see it's moving the same direction? I have no idea what I said. Okay. So I opened massive amounts of sodium channels and sodium rushed in, yes? Which brought the number maybe up to 0, okay? And now what I want to do is I want to get the number really quickly back to -70. Close those sodium channels, open massive amounts of potassium channels, and potassium will move out, right? And that will get my number fixed. But let's acknowledge that we've created a problem. Everything's in the wrong damn place, isn't it? Sodium's too high on the inside, potassium's too high on the outside, right? So how do you fix that? Yeah, you pump. You were always pumping, now you work your little pump buns off, right? You pump faster, right? Does that make sense to you guys? Okay, so whereas depolarization is generally what channels?
Sodium gated channel.
Gated sodium channels. Hyperpolarization is usually gated potassium channels. Repolarization is usually, no, not pumps. The pump does not fix the number. The pump fixes the concentrations, right? The repolarization is usually gated potassium channels, okay?
So the pump is only going to fix your positive negative? Or?
No, the pump doesn't actually impact the positive and negative because it's moving 3 and 2. It's not dramatically affecting the number, right? It will affect the number, but not very quickly, right? If you're moving positives both directions, then you're not going to. Quicker, yes. You depend on it really. You'll see why it needs to be really quick. Go ahead.
So repolarization only cares about getting the charge back into place?
Repolarization, you're not marking locations. Do you see with this electrical map? You're really not even acknowledging the source of the positive charges or negative charges. You're just looking at the number, right? So the repolarization will first fix the number and then fix the concentrations, yes? So the pump doesn't dramatically affect the number. It does affect it, but not dramatically. Did you have a question? No? Okay, so I'm going to back up for just a second, and I want to ask you to put this together. So you just told me how you changed resting membrane potential, right? Sodium and potassium channels. Here's my question. How did you get to -70 in the first place? What kind of channels were involved in getting to -70? It's leak channels, yes. So leak channels get you to resting membrane potentials. Gated channels change it, right? It was the unequal leaking of sodium and potassium that got you to -70. So leak channels to get here, gated sodium channels here, gated potassium channels here, and gated potassium channels here. Is that okay? Okay, now back up one more time. For those of you who care, this doesn't have to be a sodium channel like we said, and it can be any positive ion that would move in, right? Or for those of you who care about such things, any negative ion that moved out. Okay, likewise here, this could be any positive ion that moves out for hyperpolarization or any negative ion that moves in. Do the same thing. Now, we don't run into those examples very often because they make students want to throw things at me. So most of the time,
Let's just stick with that.
well, I mean, but you know, there are some people who like are so logic brain, they're like, what's special about sodium? Well, there's nothing special about sodium. It's just that we know what it's going to do because we worked really hard on it, okay? But do not get surprised later on when you, for instance, run into cardiac muscle that instead of using sodium for depolarization, uses calcium for depolarization, right? Because it's a positive ion and it's going to move in. There's nothing that said that you couldn't. It's just that usually it's going to be sodium and potassium. Okay, everybody all right? Okay, so next time what we are going to do is we are going to start talking about the types of changes that you do. So we did depolarization and hyperpolarization. You can either do little bitty changes or you can do, here's a small change. Okay, let me show you. So this is a very small change in membrane potential. If this is -70, because they didn't mark it, is this a depolarizing or a hyperpolarizing change? That's depolarizing. Or you could do a massive change, and this massive change is going to be an action potential. This is a graded potential, small, diminishes as you get further away from it. This is an action potential, massive, okay? Only electrically active tissues do this because it actually is going to involve voltage-gated channels. Okay, so you have a quiz next time I see you.
Since I'm in the other class, do I have to come to the other class to take the quiz?
Yes.
Did you say hyperpolarization, they're more electrically the same?
Different.
And then depolarization is polarized.
Yes, because polarized means different, right? And if you depolarize, you make them less different. And if you hyperpolarize, you make them more different, right?